What is a chromosome? In previous animations, we learned that DNA is a spiraling chain-like molecule made up of 4 different types of nucleotides. Scientists have labeled them A, C, T, and G. We also learned that a gene is a long stretch of DNA, a specific sequence of A's, C's, T's, and G's that code for something. What does a gene code for? Usually a protein or a group of proteins, though there are a few exceptions. In multicelled creatures like ourselves, proteins, which are coded for by genes, interact with other proteins and molecules to make up living cells. Cells make up tissues, tissues make up organs, and organs ultimately make up entire living creatures. Individuals have different traits, in this case differences in beak shape, largely because of differences or mutations in their genetic code. These differences can either cause changes in a specific protein shape, and therefore its function, or mutations can change when or how much of a particular protein will be produced. Now that you clearly understand these 3 terms, it is now time to talk about chromosomes. A chromosome is an entire chain of DNA along with a group of stabilizing proteins. You may have seen images showing DNA trapped inside a chromosome, sort of like this. This is not how chromosomes hold their DNA. If you were to unravel a chromosome, you would see that it's made of a thread, sort of like a spool of yarn. Zoom in again and you'll see that that fiber is a collection of protein orbs called histones, which are wrapped with a string-like structure. Zoom in on that string and you'll find that this is the chain of DNA. It is extremely long, millions of nucleotides long, and often contains hundreds of genes along its length. In this image, we're looking at a chromosome in its condensed, yarn-like form, but chromosomes are only packaged tightly like this when a cell is reproducing. This structure helps with durability as the cell is splitting in 2. For most of a cell's life, however, chromosomes exist in a loose, noodle-like structure along with other chromosomes safe inside the nucleus or centerpiece of a cell. Here is a photograph of an entire set of human chromosomes taken from a cell right as it was getting ready to reproduce. Your genome, the entire collection of genes that help make you who you are written across 23 pairs of chromosomes. That's 46 altogether. With very few exceptions, each cell in your body contains its own full copy of your chromosomes, your entire genetic code. Eyeball cells only use the eyeball cell genes, the rest are turned off. Liver cells only use liver cell genes, the rest are turned off, and so on for each cell type. One member of each pair of your chromosomes came from your mother, the other came from your father. If you were to examine the 2 individuals of a pair, you'd find that for the most part, both contain the exact same genes in the exact same locations. If you were to zoom into the actual genetic code, however, you would find slight sequence variations between them. These are the results of mutations. Most of these mutations happened long ago and have been passed down from parent to child for many generations. Others, however, are completely unique to you. These mutations occurred either in the sperm or egg, which fused to make you, or they occurred early on in your development, making it into all or many of your cells as you matured. These unique mutations represent brand new genetic information, sequences of DNA that may have never been seen before in the history of the cosmos. So, to sum things up, what exactly is a chromosome? A chromosome is an entire chain of DNA along with a group of stabilizing proteins. Humans typically have 23 pairs of chromosomes in each of their cells. That's 46 altogether. In your body, one member of each pair came from your father, the other from your mother.
What are telomeres? Telomeres are structures made from DNA sequences and proteins found at the ends of chromosomes. They cap and protect the end of a chromosome like the end of a shoelace. In almost all animals, from the simplest to the most complex, telomeres are required for cell division. With each cell replication, the telomeres get shorter and shorter until they're so short that your cells can no longer divide. When cells no longer divide, tissues age. However, telomeres can be rebuilt by an enzyme called telomerase to restore cell division. If you think we've found the fountain of youth, think again. If a cell keeps dividing uncontrollably and overcomes the limitations of telomeres, a cancerous tumor can form. This is why telomeres are so important. In healthy cells, they achieve the necessary balance between limiting cellular lifespan and your own longevity and keeping cancer growth at bay.
The Human Genome Project was the first attempt to map our genetic makeup. It began in 1990 and it ended, and the announcements were made in 2003. Before the Human Genome Project, there was a belief in the life sciences, in biology, that it took one gene to program each protein in the human body. And because there are approximately 100,000 proteins, give or take, the thinking was, if it takes one gene to create each of those, that we would have about 100,000 genes. And the implications for that were vast. The pharmaceutical companies believed that if they could isolate those 100,000 genes, that they could create products that could target each of those individual genes. There's 100,000 new products. And in terms of disease and what it means for longevity and for, everything from cancers to diabetes to, whatever is happening in the human body, the thinking was it could be addressed through understanding the map of these genes. What happened surprised everyone, because at the end of the Genome Project, it was discovered that the human body only has about 20 to 25,000 genes. That's not just a little mistake. That's not just a small margin of error. That is a magnitude, several magnitudes of difference from what was believed to be happening. And what it told the researchers was that something about their theory was wrong. And what they discovered is that it's not a one-to-one correspondence. It's not one gene for every protein. Individual genes have the ability to program different proteins. This is where epigenetics comes from. It is the environment that determines how the gene expresses and what proteins it creates. Some genes can create many, many, many proteins. And when I say environment, it can be the external environment or the world around us. So, those are toxins in the environment can downregulate genes, and harmful UV radiation or radiation from a nuclear power plant or, those kinds of things. But perhaps the most powerful environment is not outside of us, it's inside of us. It is the thoughts, feelings, emotions, beliefs. Stress is a powerful epigenetic factor that can upregulate or downregulate these genes. So with only 20 to 25,000 genes, we can program that 100,000 plus a number of proteins that allows us to be who we are. That was a big deal, and scientists don't like to talk about that very much because it means everything they were teaching up until 2003 was based on this one gene for one protein principle. It was wrong. It was wrong, and it was hard for them to admit that. And it opened the door to a powerful possibility of self-regulation. This is something that comes into play for a lot of advocates when it comes to Darwin's theory. So I have no problem with the theory of evolution to explain certain forms of life. Here's where the issue comes in. Darwin's theory of evolution breaks down when it comes to humans. When it comes to us, scientists are pretty much in agreement, well, they were in agreement, that we appeared upon this earth about 200,000 years ago. A recent discovery has now pushed that date back to about 300,000 years. No controversy around that. The question is, how did it happen? They are in agreement that what are called anatomically modern humans, that's us, and the acronym is AMH. Anatomically modern humans appeared 200 to 300,000 years ago. The issue comes in terms of when we appeared and the way we appeared. We appeared relatively suddenly in the overall scheme of things. We appeared with a brain capacity, a brain size, 50% larger than our nearest primate relative. We appeared with the ability for complex speech in a way that no other form of life and no other primate has. And These capabilities are due to mutations that happened in our genome in a way that cannot be attributed to natural processes. They cannot be attributed to evolution, at least as we know evolution today. Evolution through Darwin's perspective, or from Darwin's perspective, is slow change gradually over long periods of time. Stephen Jay Gould, another evolutionary biologist, put forth the theory of what's called punctuated evolution, where it looks like things aren't happening for a while and then there's a big jump and then nothing happens in the big jump. We don't really fit into any of those, and I want to talk to you a little bit about why that is, and it has to do with the genome. In the human genome, one of our chromosomes is a mysterious chromosome. It's number 2, and it provides a large percentage, I don't know the exact percentage, but a large percentage of the DNA in our nucleus of our genome is this it's the second largest chromosome in the human body. I know that. How chromosome 2 came to be is controversial, and it is mysterious. But let me tell you why it's important. There are about 12 to 1,300 genes on human chromosome number 2. I'm going to talk about only one of those genes. It's called TBR2. TBR2 is responsible for the brain size, twice as large as our nearest primate relative, is chimps and gorillas and apes, twice as large. It is responsible for about 86% of the neocortex. And the neocortex in the human brain is where we attribute so many of the faculties that give us our humanness, our ability for empathy and sympathy and compassion, our ability to self-regulate our own biology. And so much of our mirror neurons, for example, are in the neocortex. Gene TBR1 is responsible for much of that. And gene TBR1 is the product of something mysterious that happened to create human chromosome number 2. If we didn't have chromosome number 2, we wouldn't be here. Now, what it is that happened to create human chromosome 2, this is where the mystery comes in. You've probably heard of telomeres. Some people pronounce them telomeres. The protective caps on the end of the chromosomes, so that each time a cell divides, that's a traumatic experience for the chromosome. And the telomeres take the hit of that trauma rather than the vital DNA information below the telomeres. So if you're going to have a cell division and a part of the DNA does not make that split, you don't want that to be a vital piece of information that programs your immune system or your circulatory system or something like that. So the way that we are made is that the telomeres will be the portions that don't make the cut, and all the vital DNA inside does. Those telomeres are on the ends of the chromosomes. So I'm saying this to you because in human chromosome 2, not only are telomeres on the ends of the chromosomes, they're also in the middle. And they are in the middle at a pinch point where scientists now know that 2 previously existing fully intact chromosomes fused together. So 2 pre-existing chromosomes fused together, and the fusion where the telomeres used to be on the ends, they're now in the middle. But that's not where it ends. After that fusion occurred, there were genes that were silenced, is a term that's used when the genes are no longer active. There were genes that appeared to be removed, genes that appeared to be inserted to stabilize the fusion and to take away the redundancy. When you think about this, if there were 2 fully intact chromosomes, they were each doing their own thing, and all the functions were there. When you fuse them together, there is an overlap in function. There's a redundancy, and you don't want that. You don't want them working against one another. So there was a process where certain genes were either removed or they were silenced, or some others were activated. All of this happened in a very short period of time. Now, we know of nothing in nature that provides or allows for this kind of a fusion. Now, I want to be really clear on this because I know biologists are watching this. We do see a fusion of chromosomes in nature. And it's actually more common than you would think. However, the way that happens is a chromosome. So here, this pin is a chromosome, and the telomeres are the shiny places on the end. And every once in a while, something will happen, and a chromosome will break in nature. It could be high UV radiation. It could be environmental toxins. So there are times where human chromosomes will break. And what happens is when they break, they will fuse at those break points. So the telomeres are still on the ends. There's no telomeres in the middle, and they'll fuse at those break points. That's not what happened to you and me. What is responsible for us, one of the things, is that there was the pre-existing, fully functional chromosomes that fused and then were modified after the fusion in a way that cannot be explained through natural processes. All right, that's one thing. Now, as strange as that is, if that was the only thing that happened, or as rare as that is, you could say, wow, that it's a fluke, but it happened. It's not the only mutation that happened. At the same time, there was another mutation that was happening in chromosome number 7. The result of that mutation gave us the ability for complex speech. It's the reason that chimpanzees cannot sing, and apes and gorillas cannot sing, but humans do. For over 75 million years, this chromosome, these genes in chromosome 7, they were stable, all right? And all of a sudden, there was a little, just this little switch in the DNA letters. You've seen the letters in what's called the genetic alphabet, C, T, A, G, that are abbreviations for the components that make up the DNA. There was a little switch in 2 of those letters that connected our tongue and the muscles in our jaw and the part of our brain that allows for complex speech and singing that happened only to us. It didn't happen to any other mammals, no other primates. It only happened to humans. And here's the kicker. It happened at exactly the same time. At 2 to 300,000 year range, when chromosome 2 was also fused together when humans emerged. So when we emerged, we were fully enabled and fully intact with the potential and the capabilities that we have today. They did not develop slowly, gradually over a long period of time. Something happened between 2 and 300,000 years ago that cannot be explained by Darwin's theory of evolution. That precipitated the emergence of the first anatomically modern human. And that something is a mystery. That something is, along with the miscalculations in the genome. Just to give you an idea, if you're a statistician, scientists say that the odds, when the odds of something occurring are 10 to the 400th power. So 10 times 10 times 10 times 10, 400 times. If the odds of something occurring are 10 to the 400th, scientifically that is said to be impossible. Those are considered to be impossible odds. If you had a lottery ticket and the chances of that lottery ticket being a winning ticket were 10 to the 400th, I wouldn't spend much time on that. Scientists have calculated now that the odds of our genome in general forming and chromosome 2, the way it is formed as part of that the odds of that happening are 10 to the 600th. If 10 to the 400th is impossible, 10 to the 600th is more than impossible. It's more than impossible that we're the product of random mutations, natural selection resulting from Darwin's theory. So the point is, if we are not the product of natural selection and random mutations, then what are we the product of? And this is what opens the door to a very empowering conversation. As a scientist, what I'm going to say to you is scientifically, we do not know. Now, we have theories, we have opinions, we have intuition. Scientifically, we do not know. And the reason we do not know is because the scientific community is reluctant to follow the evidence to the story that it tells. Rather, they are very emphatic that they want to take the new discoveries, the new evidence, and force it into a pre-existing story that is called Darwin's theory of evolution. I am adamant that something breaks down in the evolutionary story 200 to 300,000 years ago when it comes to us. We're the product of something, and as a scientist, I can only say this. It appears that we're the product of an intervention, an intelligent intervention. Some people call it a divine intervention that took the pre-existing forms of life, the most advanced pre-existing forms of life that existed at that time, and catapulted us light years beyond that. How that happened and who we attribute that to, that's a whole different that'll be another video. It's a whole different conversation, but you can see where religion comes in, because religion picks up with that intervention. Religion picks up with some kind of what is called a divine intervention